crime scenes are messy. These students in Boston University's Biomedical Forensic Sciences program are learning how to make sense of them. Specifically, they're studying the art and science of bloodstain pattern analysis.
Bloodstain evidence is critical. I've had one case where one single spatter of blood made the case.
But new research suggests that some models detectives are using to interpret bloodstains may be out of date, leading to faulty conclusions about how a crime was committed, or worse, incorrect verdicts in the courtroom. Now, specialists in fluid mechanics are stepping in to help crime scene investigators better understand the way blood behaves on different surfaces, with potentially game-changing implications for the criminal justice system. Detectives have been using bloodstains to help solve crimes since crime solving began, and bloodstain pattern analysis has been a serious forensic discipline for at least 100 years.
What's going to be critical is going to be the length and width of your stains when we're looking at impact patterns.
Ken Martin was with the Massachusetts State Police for 33 years, and he's been teaching bloodstain pattern analysis since 2005.
Many serious crimes, such as homicides, involve the letting or the shedding of blood. First thing you want to do is identify the patterns, and then once you identify the patterns, we expect certain things to happen. Blood being a fluid has to follow the laws of physics and fluid dynamics, so I know that if an impact spatter occurs, this will happen.
But what exactly is happening when a bloodstain forms? This is a single drop of blood falling vertically through the air, about to make contact with a hard surface. As the blood falls, cohesive forces between the molecules cause it to coalesce into a sphere, which carries momentum downward due to the pull of gravity. When it lands, the impact creates pressure on the drop, which diverts its momentum outward, causing the liquid to spread rapidly across the surface. Eventually, those same cohesive forces between the molecules stop the blood from spreading outward, and will actually try to pull it back into a sphere. What prevents this is a phenomenon called pinning. When the blood stops spreading, microscopic irregularities in the surface will latch onto the edge of the fluid, effectively pinning the edge to the surface. Where the edge gets pinned will ultimately form the perimeter of the stain when the blood dries. Because blood obeys the laws of physics, detectives can be confident that it will always behave this way, given similar conditions. Except that sometimes it doesn't.
Here I could have an identical surface in a kitchen, and on one portion of the floor the blood's adhering as we would anticipate, but in another section the blood is literally peeling up. So the question arises, why is it doing it here and not here? That, along with the drying of blood, has always been an interest area where I've thought that we could use a lot of research.
Luckily, across the Boston University campus, a lab in the mechanical engineering department is trying to answer just that question.
A lot of the experiments people have done looking at bloodstains have been on clean surfaces. Well, most surfaces aren't clean.
J.C. Bird is a specialist in fluid mechanics at Boston University. Bird's lab studies the subtle and surprising ways that fluids can behave at small scales, and they've done a lot of research on the behavior of liquid on hydrophobic surfaces. That is, surfaces that are uniquely good at repelling fluids.
Even your cell phone cover. Right now, often there will be anti-fingerprint or paints that are designed to be anti-graffiti. If the surface coating is playing a role, will that lead to differences in the stains that would fundamentally change how a crime scene was interpreted?
For example, here are 3 drops of water falling onto a glass surface. In each case, the glass itself is the same, but 2 of the surfaces have been treated with hydrophobic coatings. Here's what happens when a drop of water falls onto clean, untreated glass. This glass has had an acrylic coating applied to it, and this glass has been treated with an anti-fingerprint coating. It's immediately obvious that a drop of water behaves very differently depending on how the surface it impacts has been treated. But water is a very simple molecule, one of the simplest, which is what makes it so good for experiments.
With blood, it's not like that. It's a complex liquid. It's a combination of proteins and blood cells, which end up leading to a very different type of behavior.
And so, in 2020, Bird received funding from the National Institute of Justice to study the fluid mechanics of, that's right, human blood. Specifically, what happens when blood makes contact with surfaces with different hydrophobic properties.
It's a very fresh blood from the day before yesterday. I'm gonna distribute it into the small containers.
Garam Lee is a PhD student in Bird's lab. To prepare for this project, Lee studied bloodstain pattern analysis alongside Ken Martin's forensic students, and she's been conducting lab experiments with human blood for the past year.
One of the challenges that we have to do the experiment in a very short period of time after the blood arrives. So sometimes I work full day and night to finish up the experiment after it arrives.
In the Bird Lab, all research projects are part science experiment, part film shoot. Because a drop of blood only takes a few milliseconds to make contact with a surface, the lab uses studio lighting, magnifying lenses, and special high-speed cameras that can film tens of thousands of frames per second. Bird's lab is doing a range of experiments with the blood, from simulating spatter patterns associated with stab wounds to studying the dynamics of a single drop of blood falling on a surface. How important can a single drop of blood possibly be? It turns out, very important. One key question at many crime scenes is whether a victim was standing, sitting, or lying down when they were assaulted, and the size of a bloodstain is often used to answer that question. According to current models, 2 drops of blood that fall from the same height onto the same surface will leave the same size stain. But what if 2 drops of blood fall from the same height onto the same surface, but some parts of that surface are coated differently than others?
Let's say you have a wooden floor, and if you just waxed it, you might not be able to distinguish between the waxed floor and just the pure wooden floor.
For this experiment, Lee prepares a syringe to produce single drops of human blood, which she'll drop onto different pieces of wood. Bird's lab wants to demonstrate how blood behaves on identical surfaces that have been treated with different coatings. In this case, a thin layer of floor wax or a light scorching by fire.
What we're trying to articulate is that what the bulk material is somewhat irrelevant. It's really the wax or soot that the blood is interacting with, not the floor underneath it.
With the blood warmed up and the cameras rolling, Lee sets to work. As the 2 drops travel through the air, they're both behaving as you'd expect, coalescing into spheres because of that cohesive force between the molecules. It's when they contact the differently coated surfaces that things get interesting. The first drop hits the waxed wood and spreads out, but at the point where it should get pinned, it doesn't. That's because the wax has effectively filled in any irregularities on the wood that the blood would latch onto. Instead, the drop begins to recoil, trying to pull itself back into a sphere. The only thing that stops it is the adhesion force between the wood and the blood molecules. What about the burned wood? When the second drop of blood hits the burned wood, it spreads out just like the first drop, but then it recoils completely, to the point where it actually bounces off the surface of the wood. Not only did the edge not get pinned, but the soot coating from the burning process even interfered with the adhesion force between the blood and the wood, allowing the cohesion force between the blood molecules to pull it back into a sphere. Once these 2 drops of blood dry, they will result in completely different stains, even though their originating conditions were the same.
And that is something that's not accounted for in any of these current models.
While it may be too early for Bird's research to have reached the courtroom, his findings are at least making their way into the classroom.
Depending on how hydrophobic the treatment is of the surface, where the blood droplet literally bounces off the surface. I mean, that's some really interesting stuff that we weren't aware of in the past.
And so through experiments, we can see this, we can see situations where it does happen, where it doesn't, and we can try to understand the fundamental physics, the fluid mechanics that are responsible for it. Will our work make a difference in court cases and crime scenes? I certainly hope so.
Blood evidence presented in court cases can have serious impacts on people's lives. As Ken Martin said, a single drop of blood can potentially make the difference between an innocent or guilty verdict. So one thing should be abundantly clear. The evidence found at crime scenes needs to be supported by evidence derived from rigorous scientific experimentation. Without that, it's all just a bloody mess.